Hello, ladies and gentlemen. We are just about to get started here. Managed to get the theme song not looping this time. That's a classic Mark mistake. Hey guys, how's it going? Lots to talk about tonight, and I've been thinking about different ways of kind of wrapping this conversation around vaccines and immunity and understanding these biological processes a little bit better, so that when we have conversations with people, whether they're vaccine hesitant or not, just having literate conversations with people about the processes that are involved in your immune system, vaccinations, and just biology in general. I find there's a lot of ignorance in this space. So to get things kicked off, I want to start tonight's stream with a little bit of hype. We're gonna get the hype train going about some really crazy future technology. We're gonna talk about how it might completely change the world, and then we're gonna bring that energy back down with some cold hard realism. So I think we're in for an interesting roller coaster here. So we're just gonna dive in. This video was made a little while ago. AlphaFold has since gone through another iteration and has had a lot more success, but I feel like this video does a fairly good job of encapsulating the scope of the project, what they're trying to do. And again, it kind of gets us hyped up for, the possible future of what these new technologies are bringing to the table. So let's just dive in. I'm gonna try not to blow everyone's ears out with the volume here. So let's see how that goes.
We've discovered more about the world than any other civilization before us. But we have been stuck on this one problem. How do proteins fold up? How do proteins go from a string of amino acids to a compact shape that acts as a machine and drives life?
When you find out about proteins, it is very exciting.
You could think of them as little biological nanomachines.
They are essentially the fundamental building blocks that power everything living on this planet.
If we can reliably predict protein structures using AI, that could change the way we understand the natural world.
Protein folding is one of these holy grail type problems in biology. We've always hypothesized that AI should be helpful to make these kinds of big scientific breakthroughs more quickly.
And then I'll probably be looking at little tunings that might make a difference.
You should be creating a histogram one and a background histogram.
We've been working on our system AlphaFold really hard now for over 2 years. Rather than having to do painstaking experiments, in the future biologists might be able to instead rely on AI methods to directly predict structures quickly and efficiently.
Generally speaking, biologists tend to be quite skeptical of computational work, and I think that skepticism is healthy and I respect it, but I feel very excited about what AlphaFold can achieve.
CASP is when we say, look, DeepMind is doing protein folding, this is how good we are, and maybe it's better than everybody else, maybe it isn't.
We decided to enter CASP competition because it represented the Olympics of protein folding.
CASP, we started to try and speed up the solution to the protein folding problem. When we started CASP in 1994, I certainly was naive about how hard this was going to be.
It was very cumbersome to do that because it took a long time.
Let's see what are we doing still to improve.
Typically 100 different groups from around the world participate in CASP, and we take a set of 100 proteins and we ask the groups to send us what they think the structures look like.
We can reach 57.9 GDT.
Sorry, guys.
CASP 12 ground truth. CASP has a metric on which you will be scored, which is this GDT metric.
On a scale of 0 to 100, you would expect a GDT over 90 to be a solution to the problem.
If we do achieve this, this has incredible medical relevance.
The implications are immense, from how diseases progress how you can discover new drugs.
We are going to touch on some of the specifics, by the way. A friend of mine mentioned that this would be a good thing to be able to bring up, because a lot of people talk about like the hype around, oh, this could do this, it could do that, but like what specific diseases are related to either the misfolding of protein, protein deficiencies, allergies to proteins because of an overabundance of other proteins. So
It's endless.
I wanted to make a really simple system, and the results have been surprisingly good.
The team got some results with a new technique. Not only is it more accurate, but it's much faster than the old system.
I think we'll substantially exceed what we're doing right now.
Well, then it's a game changer, I think.
In CASP 13, something very significant had happened. For the first time, we saw the effective application of artificial intelligence.
We've advanced the state of the art in the field, so that's fantastic, but we've still got a long way to go before we've solved it.
The shapes were now approximately correct for many of the proteins, but the details, exactly where each atom sits, which is really what one would call a solution, were not yet there.
It doesn't help if you have the tallest ladder when you're going to the moon.
We hit a little bit of a brick wall since we won CASP. Then it was back to the drawing board and like, what are our new ideas? And then it's taken a little while, I would say, for them to get back to where they were, but with the new ideas. And then now I think we're seeing the benefits of the new ideas. They can go further, right? So that's a really important moment. We've seen that moment so many times now. But I know what that means now, and I know this is the time now to press. We need to double down and go as fast as possible from here. I think we've got no time to lose.
So the intention is...
I just want to say, I love like the intensity, like how they're ratchet, the intensity is used to get a guy running through. They're probably like, hey dude, there's like cake in the break room. Like he's probably running just to get food. There's no sense of like, I could be wrong. I could be wrong.
CASP is deeply stressful.
There's something weird going on with the learning, because it is learning something that's correlated with GDT, but it's not calibrated.
I feel slightly uncomfortable. We should be learning this, in the blink of an eye. The technology advancing outside DeepMind is also doing incredible work.
And there's always the possibility another team has come somewhere out of left field that we don't even know about.
Someone asked me, well, should we panic now? Of course, we should have been panicking before.
It does seem to do better, but still doesn't do quite as well as the best model. So it looks like there's room for improvement.
There's always a risk that you've missed something, and that's why blind assessments like CASP are so important to validate whether our results are real. Obviously, I'm excited to see how CASP 14 goes.
My expectation is we get our heads down, we focus on the full goal, which is to solve the whole problem.
The Prime Minister has announced the most drastic limits to eyes that the UK has ever seen in living memory.
I must give the British people a very simple instruction. You must stay at home.
We were prepared for CASP to start on April 15th because that's when it was originally scheduled to start, and it's been delayed by a month due to coronavirus. I really miss everyone.
I struggled a little bit just kind of getting into a routine, especially with my wife. She came down with the virus. I mean, luckily it didn't turn out too serious.
CASP started on Monday.
Can I just check this diagram you've got here, John? This one where we ask ground truth. Is this one we've done badly on?
We're actually quite good on this region. If you imagine that we hadn't have said it came around this way, but had put it in
down instead. Yeah.
One of the hardest proteins we've gotten in CASP thus far is a SARS-CoV-2 protein called ORF8.
ORF8 is a coronavirus protein. We tried really hard to improve our prediction. Like, really, really hard. Probably the most time that we have ever spent on a single target.
Wow.
So we're about 2/3 of the way through CASP, and we've gotten 3 answers back.
We now have a ground truth for ORF8, which is one of the coronavirus proteins. And it turns out we did really well in predicting that.
Amazing job, everyone, the whole team. It's been an incredible effort.
So just to be clear, what you're looking at, and we talked about this in a previous talk, where we talked specifically about AlphaFold and AlphaFold 2. So the ground truth, essentially, is the three-dimensional structure of that protein that has been determined using traditional methods like X-ray crystallography and a variety of other imaging techniques. And then you have in the blue the AI's prediction. And obviously, excuse me, you can see where it has a high level of correlation because they're both like really wrapped closely together, versus, you know, where there is deviation is where the prediction got a little bit wrong.
Here what we saw in CAS-14 was a group delivering atomic accuracy off the bat, essentially solving what in our world is two problems. How do you look to find the right solution, and then how do you recognize you've got the right solution when you're there?
All right, are we mostly here? I'm going to read an email. I got this from John Malt. I'll just read it. It says, John, as I expect you know, your group has performed amazingly well in CAS-14, both relative to other groups and in absolute model accuracy. Congratulations on this work. It is really outstanding.
AlphaFold represents a huge leap forward that I hope will really accelerate drug discovery and help us to better understand disease. It's pretty mind-blowing.
You know, these results were, for me, having worked on this problem so long, after many, many stops and starts, and will this ever get there? Suddenly, this is a solution. We'd solved the problem. This gives you such excitement about the way science works, about how you can never see exactly or even approximately what's going to happen next. There are always these surprises, and that really, as a scientist, is what keeps you going. What's going to be the next surprise?
Really cool, absolutely phenomenal technology, and like a huge breakthrough. So obviously there's the AlphaFold 2 project, which is, you know, even has made even better predictions. So that's where the hype starts. Did this specifically contribute any of the ammo? This is for future uses. This is for, and we're gonna get a little bit deeper into the actual technicals. There's a really good talk. Like I said, we're starting the hype train with like, oh my God, this stuff's gonna save the world. And then we're gonna cap it off with a video like streaming AlphaFold 2 is amazing, but protein folding is still not solved. So this will be a little bit of a critique of maybe like where we are in terms of our progress or where we have to go. And understanding a little bit about like the practical applications, or at least like the timelines around practical applications, because I think that's where it's obviously really cool to talk about, oh, you know, predictions in theory, but we need to be able to apply that stuff. So I wouldn't say it didn't have anything to do with the mRNA stuff. The interesting thing, and actually I will play, since you asked this question, we'll bring this up a video that is related to this, because it explains why, like, so if you encounter people who might say, well, why have we never used mRNA before? It's a new technology. It's by no means a new technology. Like we've known about mRNA vaccines for quite some time. The challenge with mRNA is that the actual molecules that we synthesize are very fragile and also hard to get to go into the body without being destroyed. And so the breakthrough that I think BioNTech kind of came up with, and Pfizer, you know, as a partner of theirs, is they created a lipid nanoparticle, or a tiny particle of fat, engineered lipid, that the mRNA sits inside of, and that fatty lipid that goes into your arm gets stuck to a cell, your cell takes it in, and then the mRNA is processed inside of the cell like any other piece of mRNA. So let's touch on that really quick. There's the, we can leave, Lex Friedman. Vaccine. It is right. Vaccines, modern vaccines. How does the mRNA vaccine work? Here we go.
But, you know, I still think it's very surprising that something like that works, right?
Yeah, I am surprised.
So you're just launching RNA in a protective membrane.
Yeah.
And then,
oops.
Now, one thing is surprising, that the RNA sort of lasts long enough. Right.
Also, I want to make another point too. One of the really awesome things about mRNA vaccines, we can produce them significantly quicker because of the nature of the technology. We don't actually have to culture virus like we do with traditional vaccines. It's a lot safer. There's a lot less opportunities for things like contamination. And again, just our ability to scale, produce, and create them safely, as opposed to using things like, again, in this conversation they have, they talk about when the polio vaccine was first introduced, like a bunch of kids were like paralyzed because the active polio virus hadn't been like inactivated properly. So
in its structure, but then the other thing is, why does it work that's a good training ground for the immune system? Is that obvious?
Well,
that
I don't
work?
think it's obvious to most people, and it's worth going into because it's really interesting. I mean, first of all, they wrap the RNA in fats, in lipid membranes.
There you go. Right.
And the particular formulation they test for years to make sure it's stable. You know, it lasts a long time after it's injected. And the two companies that make the current COVID vaccines, right, Moderna and Pfizer, they have different lipid formulations to get to the same. So that's a real part of it. And it's not simple. There are quite a few different lipids that they put into this coating. And they test to see how long they protect the RNA after it's injected, say, into a mouse. How long does it last? And the way it works is these lipid nanoparticles, they get injected into your muscle, they bump into cells, and they get taken up. So lipid fat is sticky. It's greasy, we like to say. And so your cells are covered with the greasy membrane also. So when these lipid nanoparticles bump into them, they stick and they eventually get taken up. And they figured this out right at the beginning. If we put RNA in a lipid nanoparticle, will it get taken up into a cell? And the answer was yes. It was just, let's try it, and it worked.
So it's basically experiment. It's not like some deep understanding of biology. It's experimentally speaking, it just seems to work.
Yeah. Well, they had some idea that lipids would target this to a cell membrane. And remember, there's no receptor involved. Like the virus has a specific protein that attaches to a receptor.
This is an important point, right? So the coronavirus spike protein connects to the what's it called, the ACE2 receptor of your cells, and it's that connection that allows it to bind and to get into your cell. Without that receptor, it can't do it. This process does not use receptors at all.
It's not efficient enough to just bump around and get into a cell. That's what these things are doing. And they probably optimize the lipids to get more efficient uptake, but it's not as efficient as a virus would be to get into a cell.
So you have no specific, I mean, which is why it's surprising that you can crack into the safe with a hammer or with some fat. I mean, that's kind of surprising. It's kind of amazing that it works. But so maybe let's try to talk about this. So one of the hesitancies around vaccines,
this is good to get into real quick,
technology, is the fact that mRNA is a new idea, and it's an idea that was shrouded in some skepticism, as you said, by the scientific community, because it's like it's a cool new technology, surprising that it works. What's your intuition? I think one nice way to approach this is try to play devil's advocate and say both sides. One side is why your intuition says that it's safe for humans, and what arguments can you see, if you could steelman an argument why it's unsafe for humans, or not unsafe for humans, but the hesitancy to take an mRNA vaccine is justified.
So many people are afraid because it's new technology and they feel it hasn't been tested. I mean, in theory, what could go wrong? This is the nice thing about mRNA is that it doesn't last forever, as opposed to DNA, which doesn't last forever, but can last a lot longer.
So this is a really good point, right? When you have live virus that's being, or not live virus, or inactivated virus being injected, those vaccines tend to last longer in your bloodstream, as opposed to the mRNA molecules just degrade naturally very quickly.
And it could even go into your DNA. Right?
Anyway, so we can come back to this, but I don't want to get too off the rails in terms of the the mRNA safety aspect of things yet. So we quickly touched on AlphaFold and why protein folding is important. So I think it's good to watch a quick video that I found that's actually kind of cool on DNA to protein in 3D. And this is going to help us better understand what's going on with vaccines, right? How is that spike protein being created? What is it being created out of? So that we actually can understand like the real mechanisms that are at play here.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA.
Just to be clear, there's like some interesting processes involved here. There's actually, this is very similar to how packets are actually sent across networks. Packets have like a header and a footer, and the same thing happens when a protein is being, or when the DNA is transcribing into messenger RNA. It's given instructions where to start and stop that instruction in like a packet size.
This process is called transcription.
Getting an A on my biology test, guys.
Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA.
Whenever you guys hear amino acids, a great, if anyone has ever seen Jurassic Park, the original Jurassic Park, you guys might remember the lysine deficiency, that all of the animals in the park are bred with a deficiency of the amino acid lysine, and if it's not fed to them, they just straight up die. So when you think here amino acids, think lysine from Jurassic Park.
Molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
And this is the process that we don't fully understand, and that AlphaFold is trying to uncover and figure out with a degree of accuracy. And just the protein, the shape of the protein determines its function. So obviously the folding determines the shape, and that process is incredibly critical. So, and again, this is an example of what, you know, these proteins look like in like a molecular model, right? So like things like actin, like this is a folding of all of these amino acids together to form these incredibly complex molecules. So to bring this all back to the immune system really quickly, we're going to just jump in and out of this video very quickly and talk about how the dendritic cells pick up parts of pathogens and get T cells going. And T cells have those specific weaponized proteins that are meant for fighting diseases. And the connection that we're trying to make back here is that if you can synthesize, or if you know how to make certain proteins or create them or trigger them, all of a sudden you have a greater control over your ability to fight infections, disease, etc. So,
of the dendritic cell, your immune system's intelligence officer. While your soldiers were bashing in heads, it was collecting samples by ripping bacteria into tiny parts and covering itself in it. Like a soldier decorating itself in the guts of a dead enemy. The cell leaves the battlefield and enters the superhighway of your immune system that connects all your tissues with your immune headquarters, your lymph nodes. The dendritic cell coming from the battlefield is looking for a helper T cell, which is a sort of all-purpose commander cell within your immune army. But not any helper T cell, one that happens to have just the right weapon for the bacteria that infected your wound. So it goes around and rubs itself, still covered in bacteria parts, against every helper T cell it meets. Most T cells are a bit disgusted and not interested, but after a few hours, something clicks. A helper T cell recognizes the bacteria parts.
So the antigen is what vaccines produce and are supposed to trigger this response. The antigen is the spike protein, or is the particular thing that this T cell is looking for.
This cell is the weapon that's needed right now. The dendritic cell is overjoyed and activates the helper T cell. Okay, wait. How come your immune system has a cell that has a weapon against the specific bacteria that infected you? Well, your immune system has a perfect weapon against every possible disease in the universe, against the Black Death, the coronavirus, or an infection that will emerge in 100 years on Mars. We'll talk about this a bit more in the next video because it's very complex, so for now, just know that you have billions of unique helper T cells that each have weapons against every possible enemy. After the right T cell is activated, your second line of defense awakes and rises like a teenager that needs to get up on a school day, very slowly. Your heavy weapons are incredibly effective, but they're not fast.
So we're gonna skip ahead a little bit because this process is like what is super critical in vaccinations, right, is creating these T cells. And this happens typically when the infection has been like sort of beaten off. So I think
about a week after you injured yourself and bacteria invaded, your second line of defense finally arrives in full force. The tiny army begins to saturate the battlefield, pinching and stunning desperate bacteria. The antibodies clump them together and make them unable to move or fight, while your soldiers massacre the defenseless victims. The tide is turning fast. As the last enemies are cleaned up, your soldiers realize they are no longer needed and begin to kill themselves to save resources. But not all of them. A few helper T cells remain and turn into memory cells.
Boom. This is the critical piece, right? Is once you receive a vaccine, you want there to be memory cells that are then stored. So if you get exposed to that pathogen, your body has the particular weapons and immunity to go after it again. This is the most important crux of
They will guard the
point I'm
for
trying to
years,
make here.
making sure the same bacteria will never again gain a foothold here. Similarly, a few B cells will stay alive and keep producing a low amount of antibodies, making you immune against this bacteria, maybe for the rest of your life. One day you wake
Which
up...
is crazy because we don't actually think about like all the things that we are actually immune to, like pathogens that we get exposed to on a regular basis that don't wreak havoc on our bodies because we have learned to some capacity to be able to fight them off, which is absolutely fascinating. So we're going to go through this video very quickly on how mRNA vaccines work. It's going to touch on a lot of the other stuff that we've talked about up to this point, but I think it does a nice job of starting to like tie it all together.
Developing vaccines is usually a very lengthy process, taking on average 8 years to get approved. Since a few years, we have the ability to create mRNA vaccines, which can be developed much faster. So how do these vaccines work? Are they safe? and how do they compare to traditional ones. But first you must understand how the immune system works. When a virus enters your body, it will attach itself to one of your cells and inject its DNA or RNA into it. This is like a blueprint for your cells, containing instructions on what the cell has to make. So in this case, the virus's RNA will tell your cell to make more copies of the same virus. They become factories, assembling new copies of the virus that can infect even more cells. But don't worry, our bodies have a defense system for foreign intruders. The immune system attacks any protein, virus, or bacteria that does not belong in our bodies, but it takes a few days for it to learn how to attack the intruder. Meanwhile, the virus factories are running non-stop, quickly replicating the virus and spreading it in your body. In other words, you start experiencing symptoms of whatever has infected you. After a few days, however, your immune system will have figured out how to attack the virus and will start producing antibodies. These attach themselves to the virus, preventing them from infecting more cells and marking them for destruction. As you can see, the immune system is remarkable, but it's slow to mount an attack. That is the reason why we can get sick in the first place. So to give it a helping hand, we developed vaccines. The main idea is to train your immune system to recognize and fight off an infection before it has occurred. Almost like showing your immune system a mugshot of the virus and saying, if you see this, kill it. There are various types of vaccines, but let's take a look at the new kid on the block, mRNA vaccines. To understand how they work, let's take the COVID-19 pandemic as an example. You might have seen pictures of the virus with its distinctive spikes. These spikes allow the virus to attach to specific cells in your body and infect them. Now, here's the key idea for the COVID-19 vaccine. What if we could train our immune system to recognize these spikes by having our own bodies produce them? To do that, researchers took the virus's blueprint, its RNA, and isolated the part responsible for producing the spikes. Armed with this blueprint, they created mRNA, or messenger RNA. This is a special form of RNA that can enter your cells and give them instructions. In this case, the RNA contains instructions to build the spikes of the coronavirus. Not the virus itself, just the spikes. So mRNA vaccines contain instructions.
And this is the important part, right? Is that the virus as a whole is what makes you sick and has the capacity to do damage to cells and to replicate. The spike protein, and there is some like credence, I will say loosely, to people be like, well, the spike protein can cause issues, it can cause inflammation, it can cause, and that is true in some cases. Like there is myocarditis and a couple of other things that people have gotten as a result of what do they call fusion? I think that's what it's referred to as when the protein, the spike protein, will actually fuse to cells and cause damage or inflammation. Apparently they edited the spike protein specifically to avoid fusion, but I'm sure that that's not like 100% infallible. So it's just a very important distinction to make. If you ever have a conversation with someone who's confused about the nature of vaccines or think that particularly the mRNA has any part of the virus in it, it doesn't, right? It's like saying, I'm not even going to come up with an analogy. I'm just going to let them keep explaining it because I'm going to say something really stupid, I'm sure.
Instructions for your cells that tell them to build a part of a virus in large volumes, almost like giving them a recipe to follow. Once this is happening, your immune system kicks into action and starts learning how to attack these intruders. Again, it takes some time for the immune system to fight off these spikes, but you won't get sick because it's only the spikes, not the virus itself. And that's it. Your immune system has learned how to attack the spikes of the coronavirus. It destroys all the spikes and even breaks down the mRNA vaccine itself. The only thing left in your body are special B cells or memory cells. These can linger around for months or years until the same virus infects you again. When that happens, the B cells can produce the correct antibodies right away, preventing the virus from spreading and making you sick. What's interesting about this mRNA technique is that it's relatively quick to develop a vaccine as soon as we know the DNA or RNA sequence of a virus. And secondly, because the vaccine only makes our bodies produce a tiny part of a virus, we cannot get sick from it. More traditional vaccines use weakened versions of the actual virus. This also triggers an immune response, but could also give you mild symptoms. Now you know how mRNA vaccines work. But don't take my word for it. All sources used to make this video are listed in the description.
So the exciting stuff that I kind of wanted to tie together before I let Lex, and we will talk a little bit about what these the ability to solve some of these protein problems and to be able to deliver designer proteins via mRNA vaccines are incredibly exciting. Like when you look at diseases like, so one of the things is the misfolding of proteins. And that process typically is involved in diseases like Alzheimer's, Parkinson's, what else? I took some notes here. Cystic fibrosis, Creutzfeldt-Jakob disease. You have missing protein disorders like protein S deficiency, which causes like serious blood clotting problems. And you can have also intolerances to proteins as well. So the connection to take something as revolutionary as AlphaFold and sort of unlock the world of proteins so that we can start designing them, delivering them through technologies like mRNA, could really change the world and bring a lot of amazing medicine to the table. There's certain tumors that we could target with specific proteins. There's a ton of diseases that we could immediately probably start treating and having a huge effect in. It really would be incredible. And so I think the technology is incredibly promising, and now I'm going to let these guys kind of poo-poo all over it. Well, I actually haven't seen this video, so we'll see what the critique is like together.
So speaking of AlphaFold, so DeepMind's AlphaFold 2 recently was announced to have quote-unquote solved protein folding. How exciting is this to you? It seems to be one of the exciting things that have happened in 2020. It's an incredible accomplishment from the looks of it. What part of it is amazing to you? What part would you say is overhyped or maybe misunderstood?
It's definitely a very exciting achievement. To give you a little bit of perspective, right? So in bioinformatics, we have several competitions, and so the way you often hear how those competitions have been explained to sort of to non-bioinformaticians is that they call it bioinformatics Olympic Games, and there are several disciplines, right? So the historically one of the first one was the discipline in predicting the protein structure, predicting the 3D coordinates of the protein. But there are some others, so the predicting protein functions, predicting effects of mutations on protein functions, then predicting protein-protein interactions. So the original one was CASP, or critical assessment of protein structure. Typically what happens during these competitions is experimental scientists solve the structures, but don't put them into the protein data bank, which is the centralized database that contains all these 3D coordinates. Instead, they hold it and release protein sequences. And now the challenge of the community is to predict the 3D structures of these proteins and then use the experimentally solved structures to assess which one is the closest one. Right.
And this competition, by the way, is a bunch of different tangents. And maybe you can also say what is protein folding. And this competition, CASP competition, has become the gold standard, and that's what was used to say that protein folding was solved. Sorry, just to add a little, just a bunch. So if you could, whenever you say stuff, maybe throw in some of the basics for the folks that might be outside of the field. Anyway, sorry.
So the reason it's relevant to our understanding of protein folding is because we've yet to learn how the folding mechanistically works, right? So there are different hypotheses what happens to this fold. For example,
so this is a really good point, is we are predicting the structures of the protein so far. We haven't figured out how it's actually done. Now, I think where the optimism lies here is that because we have some tools to do predicting where it ends up, making the leap to the how is going to be easier, because we'll start to be able to link different models and build AIs to amalgamate those models and come to conclusions using those extended dimensions of information. So I understand the optimism. This is a really good point. Is like we're definitely not there yet.
There is a hypothesis that the folding happens by, also in the modular fashion, right? So that we have protein domains that get folded independently because their structure is stable, and then the whole protein structure gets formed. But within those domains, we also have so-called secondary structure, the small alpha helices, beta sheets. So these are elements that are structurally stable, and so and the question is, when do they get formed? Because some of the secondary structure elements, you have to have a fragment in the beginning and say the fragment in the middle, right? So you cannot potentially start having the full fold from the get-go, right? So it's still,
there's a multi-part process to it.
What happens? We know that it's an extremely efficient and stable process, right?
So there's this long sequence, and the fold happens really quickly.
Exactly.
So that's really weird, right? And it happens like the same way almost every time.
Exactly. Right?
Yeah, that's the interesting thing about the mechanism, is that although it's like imagine origami. I think origami is a great way to look at this. So you have like an origami crane, an origami pig, an origami whatever. And imagine just having a piece of paper and just like it folds itself into origami. Like that's kind of what's happening here. It's like we have no idea how this paper folds itself into this origami so efficiently, and that process seems to make many different kinds of origami, not just one particular shape. So that's kind of where the magic is here.
Right?
Really weird. That's freaking weird.
It's that's why it's such an amazing thing. But most importantly, right? So it's so when you see the translation process, right? So when you don't have the whole protein translated, right? It's still being translated, getting out from the ribosome. You already see some structural fragmentation. So folding starts happening before the whole protein gets produced, right? And so this is obviously one of the biggest questions in modern molecular biology.
Not like maybe what happens, like that's bigger than the question of folding. That's the question of like deeper fundamental idea of folding.
Yes,
behind folding.
Exactly. So so obviously, if we are able to predict the end product of protein folding, we are one step closer to understanding sort of the mechanics of the protein folding, because we can then potentially look and start probing what are the critical parts of this process and what are not so critical parts of this process. So we can start decomposing this, so in a way, this protein structure prediction algorithm can be used as a tool, right? So you change the you modify the protein, you get back to this tool, it predicts, okay, it's completely unstable.
Yeah, which aspects of the input will have a big impact on the output.
Exactly. So what happens is, we typically have some sort of incremental advancement. Each stage of this CASP competition, you have groups with incremental advancement, and historically, the top performing groups were, they were not using machine learning. They were using very advanced biophysics combined with bioinformatics, combined with the data mining, and that was that would enable them to obtain protein structures of those proteins that don't have any structurally solved relatives. Because if we have another protein, say the same protein but coming from a different species, we could potentially derive some ideas, and that's so-called homology or comparative modeling, where we'll derive some ideas from the previously known structures, and that would help us tremendously in, reconstructing the 3D structure overall. But what happens when we don't have these relatives? This is when it becomes really, really hard, right? So that's so-called de novo, de novo protein structure prediction. And in this case, those methods were traditionally very good. But what happened in the last year, the original AlphaFold came into, and all of a sudden, it's much better than everyone else.
This is 2018.
Yeah.
Oh, and the competition is only every 2 years. I think.
And then, so it was sort of kind of a shockwave to the bioinformatics community that we have like a state-of-the-art machine learning system that does structure prediction, and essentially what it does, if you look at this, it actually predicts the context. So the process of reconstructing the 3D structure starts by predicting the context between the different parts of the protein, and the context essentially is the parts of the proteins that are in a close proximity to each other.
Right. So it actually, the machine learning part seems to be estimating. You can correct me if I'm wrong here, but it seems to be estimating the distance matrix, which is like the distance between the different parts.
Yeah. So we call it the contact map.
Contact map.
Right. So once you have the contact map, the reconstruction is becoming more straightforward.
Yeah.
Right. But so the contact map is the key, and so that what happened, and now we started seeing in this current stage, right? Where in the
just to take a pause here really quickly.
The emergence of
I am making an assumption here, but I would assume by contact map that's some kind of mapping out about how the different molecules and atoms in those proteins interact. The electrons, et cetera, understanding those interactions, and then being able to build a picture of the prediction.
Of this
it's my assumption.
Ideas in others people works, right? But yet here's AlphaFold 2 that's again outperforms everyone else. So by introducing yet another wave of the machine learning ideas.
Yeah, there does seem to be also an incorporation. First of all, the paper is not out yet, but there's a bunch of ideas already out. There does seem to be an incorporation of this other thing. I don't know if it's something that you could speak to, which is like the incorporation of like other structures, like evolutionary similar.
Yes.
Structures that are used to kind of give you hints.
Yes. So evolutionary similarity is something that we can detect at different levels, right? So we know, for example, that the structure of proteins is more conserved than the sequence. The sequence could be very different, but the structural shape is actually still very conserved. So that's sort of the intrinsic property that in a way related to protein folds, to the evolution of the protein of proteins and protein domains, etc. But we know that. I mean, there've been multiple studies, and ideally, if you have structures, you should use that information. However, sometimes we don't have this information. Instead, we have a bunch of sequences. Sequences we have a lot. Right, so we have, hundreds, thousands of different organisms sequenced, right? And by taking the same protein, but in different organisms, and aligning it, so making the corresponding positions aligned, we can actually say a lot about sort of what is conserved in this protein, and therefore structurally more stable, what is diverse in these proteins. So on top of that, we could provide sort of the information about the sort of the secondary structure of this protein, etc., etc. So this information is extremely useful, and it's already there. So So while it's tempting to do a complete ab initio, so you just have a protein sequence and nothing else, the reality is such that we are overwhelmed with this data. So why not use it?
Yeah.
And so I'm looking forward to reading this paper.
It does seem to like they've in the previous version of AlphaFold they didn't for the evolutionary similarity thing, they didn't use machine learning for that, or they used it as like the input to the entirety of the neural net, like the features derived from the similarity. It seems like there's some kind of quote unquote iterative thing where it seems to be part of the learning process is the incorporation of this evolutionary similarity.
Yeah, I don't think there is a bioarchive paper, right?
There's nothing.
No,
There's
nothing. Yeah,
there's a
so
blog post that's written by a marketing team essentially.
Yeah.
Which it has some scientific similarity probably to the actual methodology used, but it could be. It's like interpreting scripture. It could be just poetic interpretation.
It's a good way of putting it.
Work as opposed to direct connection to the work.
So now, speaking about protein folding, right? So in order to answer the question whether or not we have solved this, right?
Yeah.
So we need to go back to the beginning of our conversation, with the realization that an average protein is that typically what the CASP has been focusing on is this competition has been focusing on the single, maybe two domain proteins that are still very compact. And even those ones are extremely challenging to solve, right? But now we talk about, an average protein that has 2-3 protein domains. If you look at the proteins that are in charge of the, of the process with the neural system, right? Perhaps one of the most recently evolved sort of systems in an organism, right? All of them, well, the majority of them are highly multi-domain proteins. So they are, some of them have 5, 6, 7, and more domains, right? And we are very far away from understanding how these proteins are folded.
So the complexity of the protein matters here. The complexity of the protein modules, or the
not just that, but the different domains that it's like associated with it. So like a protein can obviously have one function when it comes to this particular part of your body, but it could have a completely different function in that part of your body.
So you're saying solved. So the definition of solved here is particularly the cast competition achieving.
They solved a very narrow question, and the true question around protein folding is obviously much broader, much more complex, and requires significantly more investigation. So they've figured out probably a very narrow answer to a very narrow question.
Human level, not human level. Achieving experimental level performance on these particular sets of proteins that have been used in these competitions.
Well, I mean, I do think that especially with regards to the AlphaFold, it is able to to solve at the near experimental level a pretty big majority of the more compact proteins, or protein domains, because again, in order to understand how the overall protein multi-domain protein fold, we do need to understand the structure of its individual domains.
I mean, unlike if you look at AlphaZero or like even Mu0, if you look at that work, it's nice. Reinforcement learning self-plane mechanisms are nice because it's all in simulation, so you can learn from just huge amounts. Like, you don't need data. Like, the problem with proteins, like the size, I forget how many 3D structures have been mapped, but the training data is very small, no matter what. It's like millions, maybe 1 or 2 million or something like that. But it's some very small number. But like, it doesn't seem like that's scalable. There has to be, I don't know, it feels like you want to somehow 10x the data or 100x the data somehow.
Yes, but we also can take advantage of homology models, right? So the models that are of very good quality because they are essentially obtained based on the evolutionary information, right? So you can, there is a potential to enhance this information and use it again to empower the training set. And I think I am actually very optimistic. I think it's been one of these sort of turning events where you have a system that is, a machine learning system that is truly better than the sort of the more conventional biophysics-based methods.
Yeah, it's a huge leap.
So, like I said, we're gonna start with the mad hype train that everything was gonna be solved with these amazing technologies, and obviously we're only part of the way there. So it's nice to have some realism at the end. And we're gonna shift gears a little bit. I want to talk about this really interesting new technology that NVIDIA has introduced, a couple of papers here, so we're just going to dive straight into it. Earlier this week, Netizens took to Twitter to express their disbelief and surprise, knowing that during the NVIDIA GTC conference, Jensen Huang did not deliver the entire keynote presentation himself. Some tech enthusiasts tweeted they were fooled by NVIDIA. So yeah, apparently they used for 14 seconds, his virtual replica spoke to the audience while introducing the CPU designer. Last week, the graphics processor company revealed in its blog post that it leveraged the power of Omniverse to pull the stunt off without anyone noticing. We gotta see the video of this. There's gotta be a video. Let's do this. 1 sec here. NVIDIA CEO video. Oof, I'm not sure what the 20 seconds are going to be. We'll see. Well, let's see if we can.
And virtually enhance has led to incredible advances and allowed our technology to contribute to advancing the basic sciences, the arts, and the industries. This last year, we introduced the second generation of RTX, a new rendering approach that fuses rasterization and programmable shading with hardware-accelerated ray tracing and artificial intelligence. This is the culmination of 10 years of research. RTX has reset computer graphics, giving developers a powerful new tool just as rasterization plateaus. Let me show you some amazing footage from games in development. The technology and artistry is amazing. We're giving the world's billion gamers an incredible reason to upgrade. The interchange of 3D. Creators are doing amazing things with Omniverse. At Foster and Partners,
here, hold on a second.
Designers in 17 locations around the world are designing buildings together in their Omniverse shared virtual space. I want
to see the actual footage. Maybe they do it here. I would love it if we could just get straight into it here, guys. Where is it?
Okay. So you're still not sure, right? I mean, honestly, they both looked kind of fake. I don't think he was breathing in the first one.
honestly, no normal person could tell if you weren't expecting it. And I think that's what's like absolutely fucked about this. So there is a really interesting. This is the mind blower here. This is what actually kind of like blows my mind in terms of the upcoming technology around facial generation. So they have like they've achieved an exponential level of improvement here. I will let the video speak for itself.
Papers with Dr. Károly Zsolnai Fehér. Today we will see how a small change to an already existing learning-based technique can result in...
So actually, before we dive into this, just really quickly, because I know we love demonstrations, we're going to this person, it does not exist. So there is a website that you can go to called This Person Does Not Exist. You can click on it, and these people are CGI generated faces, okay? And so typically these things have been really good at generating photorealistic faces via AI, but animating those or making those into video has been problematic. And this paper does a really, really good job of explaining that.
This is 2-minute papers with Dr. Károly Zsolnai Fehér. Today we will see how a small change to an already existing learning-based technique.
Get ready to have your minds blown, guys. This one's cool.
This is StarGAN 2, a technique that appeared in December of 2019. It is a neural network-based learning algorithm that is capable of synthesizing these eye-poppingly detailed images of human beings that don't even exist. This is all synthetic. It also supports a cool feature where we can give it a photo, then it embeds this image into a latent space, and in this space we can easily apply modifications to it. Okay, but what is this latent space thing? A latent space is a made-up place where we are trying to organize data in a way that similar things are close to each other. In our earlier work, we were looking to generate hundreds of variants of a material model to populate this scene. In this latent space, we can concoct all of these really cool digital material models. A link to this work is available in the video description. StarGAN uses walks in a similar latent space to create these human faces and animate them. So, let's see that. When we take a walk in the internal latent space of this technique, we can generate animations.
Also, if anyone has ever tripped balls on mushrooms, this is exactly what faces look like when you're tripping balls on mushrooms.
Walk in the internal latent space of this technique, we can generate animations. Let's see how StarGAN 2 does this. It is a true miracle that a computer can generate images like this. However, wait a second. Look closely.
Well, you have to look close so you can tell the hair is fucked.
Right here. Don't despair if not. It is hard to pin down what the exact problem is,
no,
but
it's his hair.
Easy to see that there is some sort of flickering going on. So, what is the issue? Well, the issue is that there are landmarks, for instance, the beard, which don't really or just barely move, and essentially the face is being generated under it with these constraints. The authors refer to this problem as texture sticking.
Interesting.
The AI suffers from a sticky beard, if you will. Imagine saying that
what an interesting
to someone, you would end up in a madhouse. Now, this new paper from scientists at NVIDIA promises a tiny but important architectural change. And we will see if this issue, which seems like quite a limitation, can be solved with it or not. And now, hold on to your papers and let's see the new method. Holy mother of papers. Do you see what I see?
That's pretty fucking impressive.
Beards are a thing of the past, and facial landmarks
I think it's still a little bit there, but yeah.
Fly about freely. And not only that, but the results are much smoother and more consistent to the point that it can not only generate photorealistic images of virtual humans. Come on, that is so 2020. This generates photorealistic videos of virtual humans. So, I wonder, did the new technique also inherit the generality of StarGAN 2? Let's see We know that it works on real humans, and now, paintings and art pieces, yes, excellent. And of course, cats and other animals as well. The small change that creates these beautiful results is what we call an equivariant filter design. Essentially, this ensures that final details move together in the inner thinking of the neural network. This is an excellent lesson on how a small and carefully designed architectural change can have a huge effect on the results. If we look under the hood, we see that the inner representation of the new method is completely different from its predecessor. You see, the features are indeed allowed to fly about, and the new method even seems to have invented a coordinate system of sorts to be able to move these things around. What an incredible idea! These learning algorithms are getting better and better with every published paper. Now, good news, it is only marginally more expensive to train and run than Stargate 2, and the less good news is that training these huge neural networks still requires a great deal of computation. The silver lining is that if it has been trained once, it can be run inexpensively for as long as we wish.
It's the training that takes like the computational power, obviously.
Might soon become a thing of the past, because from now on we can generate photorealistic videos of them.
So again, like this mixed with what you're seeing come out of NVIDIA and their CEO basically like throwing in fake stuff. So this brings me to this next wonderful article that I found that we're just going to read through. Social media influencer / model created from artificial intelligence lands 100 sponsorships. So with the advancement of technology, AI influencers and virtual human models are becoming the new trend. It has recently emerged as a blue chip in the advertising industry because there are no privacy scandals and no time-space restrictions with these virtual humans. In particular, the use of virtual humans seems to be gaining more momentum in the COVID-19 pandemic, where there may be restrictions on travel, limitations on the number of people gathering. So this will allow, again, this is all very insidious because it's all going to be used for corporations and advertising and marketing, but it will allow them basically to create any kind of media content for whatever types of, and I read a little bit about this in Max Tegmark's book, on the Prometheus team. We might hit that actually to close out the stream tonight. On September 10th, Bak Seung-yup, CEO of Sita Studio X, that created Rosie, the new high-rising blue chip on the advertising industry, explained, "These days, celebrities are sometimes withdrawn from dramas that they have been filmed because of school violence scandals or bullying controversy. However, virtual humans have zero scandals to worry about, nor do they have feelings to get hurt. So, like, trolls, like, wouldn't be able to, like, say, like, yo, fuck you, because, like, they're not real. You can't talk to them," which is interesting. Rosie is a virtual human that was created at Cydia Studio X last year in August. Her age will forever be forever 22, and has been keeping an active presence online as a real human since December of last year. In particular, this virtual human being gaining much attention as she appeared in an advertisement for Shinhan Life in July. According to CEO Bak Seung-yup, Rosie currently has the advertising contracts with companies and a significant amount of sponsorships. CEO Beck said, "We have advertised twice already this week alone and now have 8 exclusive contracts." And he continued to explain, "She has landed more than 100 sponsorships now, but we have not been able to process them yet." That's crazy. So obviously none of these pictures are real. They're all essentially iterated or generated by AI. So with that being said, people talk a lot about how deep fakes are a significant problem or going to be more and more of a problem, I should say, in terms of the manipulation of media, et cetera, et cetera. One thing that is like fairly promising right now is that AI is also really good at detecting them. So I'm going to put on this video, I'm going to grab my book really quickly, and we are going to wrap right after this.
This is 2-Minute Papers with Károly Zsolnai Fehér. We talked about a technique by the name face-to-face back in 2016, approximately 300 videos ago. It was able to take a video of us and transfer our gestures to a target subject. With techniques like this, it's now easier and cheaper than ever to create these deepfake videos of a target subject provided that we have enough training data, which is almost certainly the case for people who are the most high-value targets for these kinds of operations. Look here. Some of these videos are real, and some are fake. What do you think? Which is which? Well, here are the results. This one contains artifacts and is hence easy to spot, but the rest, it's tough. And it's getting tougher by the day. How many did you get right? Make sure to leave a comment below. However, don't despair, it's not all doom and gloom. Approximately a year ago, in came Face Forensics, a paper that contains a large dataset of original and manipulated video pairs. As this offered a ton of training data for real and forged videos, it became possible to train a deepfake detector. You can see it here in action, as these green to red colors showcase regions that the AI correctly thinks were tampered with. However, this follow-up paper by the name Face Forensics ++ contains not only an improved dataset, but provides many more valuable insights to help us detect these deepfake videos and even more. Let's dive in. Key insight number 1. As you've seen a minute ago, many of these deepfakes introduce imperfections, artifacts to the video. However, most videos that we watch on the internet are compressed, and the compression procedure, you have guessed right, also introduces artifacts to the video. From this, it follows that hiding these deepfake artifacts behind compressed videos sounds like a good strategy to fool humans and detector neural networks likewise, and not only that, but the paper also shows us by how much exactly. Here you see a table where each row shows the detection accuracy of previous techniques and a new proposed one, and the most interesting part is how this accuracy drops when we go from HQ to LQ, or in other words, from a high quality video to a lower quality one with more compression artifacts. Overall, we can get an 80 to 95% success rate, which is absolutely amazing. But, of course, you ask, amazing compared to what? Onwards to insight number 2. This chart shows how humans fared in deepfake detection, and as you can see, not too well. Don't forget, the 50% line means that the human guesses were as good as a coin flip, which means that they were not doing well at all. Face-to-face hovers around this ratio, and if you look at neural textures, you see that this is a technique that is extremely effective at fooling humans. And wait, what's that? For all the other techniques, we see that the gray bars are shorter, meaning that it's more difficult to find out if a video is a deepfake because its own artifacts are hidden behind the compression artifacts. But the opposite is the case for neural textures, perhaps because of its small footprint on the videos. Note that the state-of-the-art detector AI, for instance, the one proposed in this paper, does way better than these 204 human participants. This work does not only introduce a dataset, these cool insights, but also introduces a detector neural network. Now, hold on to your papers, because this detection pipeline is not only so powerful that it practically eats compressed deepfakes for breakfast, but it even tells us with remarkable accuracy which method was used to tamper with the input footage. Bravo! Now, it is of utmost importance that we let the people know about the existence of these techniques. This is what I'm trying to accomplish with this video. But that's not enough. So I also went to this year's biggest NATO conference and made sure that political and military decision makers are also informed about this topic. Last year, I went to the European Political Strategy Center with a similar goal. I was so nervous before both of these talks and spent a long time rehearsing them, which delayed a few videos here on the channel. However, because of your support on Patreon, I am in a fortunate situation where I can focus on doing what is right and what is the best for all of us and not worry about the financials all the time. I am really grateful for that. It really is a true privilege. Thank you.
Really should check out his stuff. All right, guys, we are going to wrap the tech talk portion of tonight's game. I am going to just put us on pause here for a moment, and then when we come back, we're going to come back to some Call of Duty. So thank you for those of you who have joined us for the tech talk piece. Now we're moving into just some like nerdy gamer streaming time. So be back in about 5-10, guys.
you wish to support us, make sure to click the pay